I'm going to be talking about some of the work we've been doing in cryo-EM. And this is really work that's been happening from UCT, but it represents people from all over South Africa. And the first thing I want to do is kind of give you an overview of the Electron Microscope Unit at UCT and just kind of show you the kind of stuff that we've been doing. So this is where we are. This is UCT Upper Campus, and we are just over here where the arrow shows. We are kind of at the back of campus, just in this position. And the current incarnation of the Electron Microscope Unit looks like this. Sorry, I'm just hiding that thing. There we go. Okay, it's kind of in my way. Oops, sorry. So we're in a brand new building. Well, it's not brand new anymore. It's about 5 years old, but it's been specifically designed for electron microscopy, and that's really nice. And so there's a series of these pink rooms over here, and those represent microscope rooms. So the microscopes can be housed there. It's also a sample preparation area, which is really great, an engineering workshop, and I'll talk about that a little bit just now. Some office space, communal area, and meeting room, as well as a data center for image analysis and processing, as well as remote operation of the microscopes. In these areas over here, so the final two TEM rooms, those rooms are actually really interesting because they they're in a humidity controlled area. They have electromagnetic radiation field correction, and they're sorry, this is not working. There we go. And they're completely isolated from the rest of the building, and so they are very resistant to vibration. So there's a lot of infrastructure that's now being put into position. And the instrumentation that we have a variety of different techniques that we can do and we can use. And that extends from a confocal Raman system, which can actually connect to one of our scanning electron microscopes. So we can do some correlative Raman and SEM imaging, as well as some other SEMs. So, and those have detectors like EDS detectors. We can do EBSD. So there are a variety of different techniques that can be done there. But what I'm going to be talking about today are the TEMs. So the cryo-EM that we do is single particle cryo-EM using TEM. Down here at the bottom, for cryo-EM sample preparation, we have a variety of different instruments that are accessible to users of the Electron Microscope Unit. The one is a high pressure freezer. So if you have cellular material, bacteria or eukaryotic cells that you want to freeze, you can do that using high pressure so that you don't get ice crystal formation to damage the cells. Those can then be cryo-sectioned or freeze substituted and embedded in resin. For the work that I'm going to be talking about today, I use this instrument over here, which is called a Vitrobot. And so the Vitrobot takes a sample which is a macromolecule that's in solution. So something like a virus or a protein or a protein in nucleic acid complex or something like that, and that's in solution. and that can then be rapidly cooled down so that it vitrifies. And when it vitrifies, it doesn't form ice, and then it can go into the microscope. So that's that's the instrument that is relevant for the talk today. The staff are also, of course, super important. So the staff of the Electron Microscope Unit consists of a director, 2 research officers. There's Nishita and there's me, scientific and technical officers. We also have an engineer who works on the repair and maintenance of the equipment, because it's very difficult to get equipment repaired and maintained so far away from Europe or America. So to get spare parts and things takes a long time, and the technicians are not that well trained. And the final person over here, that's Sean, and he deals with logistics, shipping, safety and security. So the role of this organization. So this is a kind of an organization. We do a variety of different things. The first is to provide access to equipment. Then there's the logistics and shipping. So that's especially with shipping samples to other facilities where we are unable to perform imaging, or there are international facilities that have high resolution imaging. We can ship to those facilities. We do user training, and that's really important. And we also have a service mode. We do formal teaching, so that's teaching into formal courses. People get credits based on the training that they've done, and that's for degree purposes. We also, from within the unit, initiate projects, and that's really important for driving our technology forward and kind of advancing the sorts of techniques that we use and the sorts of things that we do. Now, there's this quote. I think it comes from, in any case, one of these kind of investor sorts, and he says, "Well, you should make a list of all the things that you want to do. So if you have a particular role or a particular job, you make a long list. And then what you do is you identify the things that you would like to do. So the first 5 things are the things that you would like to do. And then what you must do is you must avoid at all costs anything beyond that. So in other words, the 6th thing. And the thing that we would like to avoid at all costs is this service mode. Because the service mode is really a situation where users will send samples and they're not really engaging with the samples in general. It takes a lot of staff time, it takes a lot of our effort, and it's not really something that we want to be doing. And so user training to us is super important, so that people can come along and do their own samples. We don't want to be collecting images for other people. Okay. So in terms of just a sort of a broad overview of the kind of stuff that we've been doing and that we do, these are more or less annual statistics. So about 3,000 microscope hours are provisioned to our users. We have about 300 users or so. It varies every year, of course, but that's approximately it. 80% of those come from the University of Cape Town, 6% from industry, 14% from other universities, and approximately 10 research groups currently from other African countries, and most of those are from Nigeria. About 25 students graduate annually after they've used our data. So the EM unit has made some kind of contribution to their degree. It's not necessarily the central thing that they're doing with their project, but in some cases it is. about 20 publications at UCT. We currently have over 40 EMDB depositions, 46 PDB depositions, and we're working at a rate of around about 5 international data collections per year, which is the kind of a brief overview of the kind of work that we're doing. So of the tasks that we do, of course, we do teaching, which is really important. We try and make it all hands-on. This is a student who's learning about the Fourier transform and diffraction and the way the resolution can be limited by an aperture. And we really try and make that as practical as possible. And so we get an optical transform, make a little aperture by cutting a hole in a piece of paper and kind of seeing the effect on the resulting image, those kinds of things. I've found that it's far more effective than trying to teach them lots of maths. There's also outreach, and we increasingly doing outreach. So historically, we've always done about 3 or 4 groups of schools every year. And that's in the last sort of 5 years or so. COVID's kind of threw a spanner in the works. But we're now working with schools that are unable to pay for access. So this is a kind of a first thing. And so the school kids can come, they can look to see what we do, they can see the instruments, they can see some samples being imaged, those kinds of things. And we really want to try and extend this. I think the next step is actually to take school teachers, I think, more than school pupils. We're looking at sort of like low-cost clip-on microscopes for cell phones and things like that can really kind of do a little bit of learning. But this kind of outreach, I think, is really important. Okay, we have other kinds of outreach as well. This is a project with a Kaiser Trust. And what happened over here with this project is that we took a whole series of projects. So people, you can see some proteins over here. This is a virus, various projects, kind of malaria projects, etc. And worked with some artists in the Eastern Cape who then represented these projects in terms of these tapestries. So that was quite a cool project. As a sort of a form of community outreach, of course, but also just to kind of produce these cool representations of our work. Which, yeah. Great. In terms of actual impact, 1961, the EM unit, which was very new at the time and it only formed in 1959, produced the first EM virus images in Africa. 1963, the first 3D model of a virus based on experimental data. 1989 this is with Trevor Sewell and Mike Lawrence. They produced the first EM tomography reconstruction in Africa. 2003 again, Trevor Sewell. The first negative stain 3D reconstruction in Africa. 2011, the first cryo-EM reconstruction in Africa. 2019, we started to improve resolution a bit. 2022, the first approximately 2 angstrom resolution structure. As you can see, this kind of progression over the years. Now, as a kind of a benchmark, I thought it would be an interesting exercise to kind of go through this and figure out how we can kind of benchmark this according to sort of international norms. So one way of doing that is to kind of just look to see the sort of the insight that you get from each of the reconstructions. So these are kind of historic reconstructions. This goes all the way from 2003, from the first, to kind of a time just before we started doing high resolution cryo-EM, and various reconstructions from various people's projects. And sort of the takeaway from this is that you can kind of see the arrangement of protein subunits. You can, each one of these little sort of blobs over here is a single protein subunit, and those protein subunits associate together, and so you get kind of an idea of what your protein complex looks like, but you don't really get very much more than that. Now, to sort of put this in context, what I've done over here is I've plotted the date that a particular milestone was reached by researchers in the EM unit. So in other words, that's along this axis over here. So along the x-axis along here. You can see time is moving forward from 1950 to 2020. And then I compared that particular technique that was done at UCT, or in the EM unit, with the first instance that it was done internationally. So in other words, with that first negative stain reconstruction, which is shown over here in A, that first negative stain reconstruction was done 1 year after the technique was invented. So the technique was invented in 1959 by Sidney Brenner. 1 year later, in 1960, UCT researchers were doing it. And so that's pretty good. It means that UCT researchers are really working on the cutting edge. They're a year away from the first publication of that particular technique. That's really great. The first 3D model, which is now shown in B, that's from EM data. The 3D model is now produced. You can see we're getting a little bit older. So in other words, internationally that happened in 1960, but we only did that in 1963 or so, I guess. Well, not we, researchers in the EM unit. I wasn't alive then. And so things are getting older. And even though these techniques are continue to be used, it's not that A is the last time that viruses were ever looked at, and also the techniques are still very valuable. By the time we get to about 1990, the techniques, the EM techniques, in terms of biological imaging, so in other words, this technique of kind of being able to look at these molecules, visualize them, see viruses, those kinds of things, that's now 20 years old. So it's not very good, because it would be nice to be doing the cutting edge in terms of techniques. But by the time we get to the first negative stain reconstruction, which is D, which is just before I arrived in the Electron Microscope Unit, that technique again is more or less 20 years old. The first cryo-EM reconstruction, E over here, again, it's about 20 years old. But more recently, we've been pulling back. So by the time you get to F over here, which is this approximately 2 angstrom structure, we're again talking about a technique that's 2 or 3 years old internationally. So it was so we've tried to pull back. So in other words, I've split this into 3 different zones. There's getting worse, there's maintaining, but approximately 20 years behind. And this final thing over here is about getting there. It's about finally getting to this area where we are doing things that we can honestly claim are cutting edge techniques. Now, one of the things that's happened over time, so in terms of the cause of all of this, is that there's been an improvement in institutional support within the EM unit. So that's been a kind of gradual procedure from 1959 over here, when there was a single technician who worked part-time operating microscopes, to the situation that we have at the moment with 7 members of staff in a nice building, and more instrumentation, etc. So things have improved, but you'll notice that there hasn't been a steady improvement in the techniques that we're doing in terms of the age of the techniques with this institutional support. So the institutional support is necessary. I think it's an absolute requirement, but it doesn't necessarily mean that you'll have success. There there are other factors involved. In red over here, I've plotted the age of the instruments. You always get this sort of zigzag pattern because the instruments each year get 1 year older, of course. So they kind of move up at sort of a 45-degree angle. And then as soon as you buy a new instrument, or a second-hand instrument in many cases, it kind of zigzags down again because nobody really uses the older instrument anymore. They kind of, the cutting edge instrument is now the newer one, and so you get this sort of zigzag pattern. So the closer the red line is to the to this baseline, the newer it is. And you can see that the instruments are the best instrument for doing this kind of work is about 20 years old currently. And yet we're still managing to do work that is 2 or 3 years away from the world's best. So instruments are certainly important. And of course, if you get the world's best instrument, of course, you can do the world's best work, but that's kind of tricky in our environment. So there have to be other factors. And one of the most important is collaboration with international people. And so these kinds of successes, each one of these little circles over here represents a successful collaboration with somebody who either has experience overseas or is an overseas researcher who then works with people from within the Electron Microscope Unit at UCT. So we can see various people. I mean, of course, Sidney Brenner, who won the Nobel Prize, who invented negative staining. Aaron Klug, who invented the technique for actually reconstructing proteins in 3 dimensions. Mike Lawrence, who had come from the MRC. Trevor Sewell, who had done some work at Birkbeck. He then continued to work with Helen Sable, who then assisted him in terms of learning these kinds of techniques. And more recently, it's been a really great collaboration with Diamond Light Source. And you know, these collaborations are crucial, but I think they really have to be supported with funding, and that's really the 4th element of this whole thing. So these orange bands over here are periods where there was good funding, and there was a lot of stuff that went forward. This very thick band over here is one that happened a while ago, but a lot of people who were involved in cryo-EM later on were trained through this particular program. So the first program was this Carnegie grant, which funded 23 MSCs, 9 PhDs, and 12 honors students. And there was a lot of training in cryo-EM through this project. It was driven by Trevor Sewell. The next was the START project. So many people in this meeting were actually involved in that. So that was also crucially important. And the latest is this project over here, which is funded by the Jan Zuckerberg Initiative. And we got funding for this project called cryo-EM Training for Southern Africa, which ends at the end of 2024, but we actually have a lot of funding, so we'll be able to go on for longer than that. And hopefully we'll be able to renew it, actually. And actually, while I'm thinking about it, if there's anybody interested in applying for this, or if anybody is interested in getting involved, you can go to the EMU website, which is this emu.uct.ac.za, to find out more and apply. So far, we've had some success with this training program. So the way that we're running it is that people are bringing their own samples, and we're taking them then through the process of preparing the samples, optimizing them, collecting data, and processing the data. And we actually do that on the person's own sample, on their own project. And these are all examples of protein structures that have been determined through that program. So that's been quite nice. Most of them have ended up being relatively low resolution. And that's not really so much about not having the kind of capacity to do high resolution data. It's generally about the the amount of time that we're allocating. And so people only are working on these projects for about 2 to 3 weeks, which is not really enough time to determine high resolution structures. So with this project over here, which is this 5.3 angstrom cryo-EM structure, that took a little bit longer. And so this student was involved for like another month or so, which was then enough to do this. But in general, people have other things to do and they've got their own projects to work on. And in many cases, in fact, this is actually the insight that they need. So for instance, with this project over here, it's this project with this AP205 nanoparticle. And the idea behind that was to decorate it with GP140 to use as a vaccine. And what they were really interested in is whether or not the GP140 was binding or not. And so you can see the AP205 nanoparticle over here without any GP140. And then after having kind of added the GP140 to it, you get this sort of region over here of density, which is the right size for GP140. It's the same with these MI3 nanoparticles over here. So you can see again GP140 bound to them, and you can clearly see that the GP140 is there. So what the question is not really what the structure of GP140 is, because everybody knows, or what the structure of MI3 is, because it's already been determined. They want to know whether or not their particle is bound, and where it's bound, and how it's bound, and how flexibly. And so you can often answer these things quite simply. And so we just leave the projects there then, once the projects have been answered, once the research questions have been answered. Okay. So that kind of brought us up to about 2018, and 2019 was the first cryo-EM reconstruction, high resolution cryo-EM reconstruction that was published. So a lot of other projects have been worked on, and they've subsequently been deposited and things, but this was the first publication that actually showed an EM map that you could actually build and interpret to atomic resolution. So the density itself was kind of reasonable. I think it was something like 3.4 angstroms or so. You can see the shape of side chains, and so the building is reasonable. And it was the first example of the first. So it was the first active nitrate structure that had ever been determined. So that was quite cool. That was from 2019. But we have a lot more than that now. As I said, we have about 5 collections per year. Also in 2019, well, this was determined in 2019, but not published. It hasn't been published yet. The glutamine synthetase from Plasmodium falciparum. Again, a very nice resolution. You can see the side chains, and so you can build the structure nicely. Cyanide dihydratase, which had been solved in 2018, but it was deposited, I think, last year, or maybe even earlier this year. So again, pretty nice structure. So this is an interesting project. This was in collaboration with a group that was looking at HPV. And so HPV has this kind of transition that it does. It gets cleaved by furin, and the cleavage by furin actually allows it to come into the cell. So the first step is that HPV kind of comes up to the cell, and it binds to heparin. That's kind of on the surface of the human cell. Furin then cleaves it. It then disconnects from the heparin and binds to an unknown receptor on the cell, which then allows it to get taken in. And the question here was, well, what is the transition after heparin, after furin cleavage? What happens, and what happens in terms of the affinity of heparin for the viral capsid before and after furin cleavage. And so it was a nice project because even though the resolution is quite poor, actually, it was at 4.6 angstroms eventually. And so we certainly couldn't see side chains or anything. I mean, you can see this is actually a little bit worse than 4.6 that I'm showing over here. This is probably at about 8 or so. You can really just see the positions of the alpha helices, but it doesn't matter because the change that you can see, even at moderate resolution, is you can see the conformation change. So all of the subunits, the furin cleavage happens, and then you get this kind of conformation change. And we could visualize that conformation change quite nicely. And so you get the insights that you need. The reason that this is restricted in terms of resolution, it doesn't have anything to do with the microscopy. It's because the whole viral capsid is a little bit flexible. Now there are techniques that you can use to get around this, and that's actually the next step that we'd like to do, because then you can get it to about 3 angstrom resolution and build it nicely. This was a recent project. The first structure of angiotensin converting enzyme. Of course, angiotensin converting enzyme is crucial for regulating blood pressure. And the full-length protein, well, this is the full-length soluble protein. It also has a component that is transmembrane, so it's the insoluble part. But this is the first visualization of that full-length protein. And this was an extremely challenging project because not only does angiotensin converting enzyme have these 2 domains. You can see this one kind of protein here connected by a loop with another protein. 2 of these proteins, which each have the 2 domains, then come together and they form a dimer. And that dimer is flexible. You have flexibility about this sort of linker region. So in other words, that moves as well. And each of these domains also has flexibility, so they kind of open and close like that. And so the underlying assumption of cryo-EM is that you're dealing with objects that are identical to one another. And so this really breaks down that assumption. And so quite complex computational methods need to be used in order to then account for that. This project took some time, and the person who was involved was Lava. So yeah, so some successes. The cover of EMBO, some nice articles in communications biology. So we're having some kind of success, and more projects. In terms of these kinds of enzymes, this particular class of enzymes, we found an interesting relationship previously at low resolution that showed us that the quaternary arrangement of structures of subunits within this protein had an important influence on the size of the substrate that could be converted. And so by altering the quaternary structure, you could then change the substrate specificity of this enzyme. And now, of course, we've seen these proteins at high resolution, high enough to kind of interpret them to atomic resolution. And what we see is we can actually measure the size of the binding pocket, and we get a very nice correlation between the size of the binding pocket and this parameter here, which is called helical twist, which is a measure of this quaternary structure. So this particular protein forms these filaments. Yeah, in terms of where we're going, this has become a relatively common result now, where you can sort of distinctly see side chains, and the resolution is a bit better. This is This is at about 2.2 angstrom resolution or so. And so you can get a visualization of the whole protein complex, which is nice. This is a nice big one. You can pick out an individual protein subunit, which is shown over here in red. And then if we want to look at how well it fits in terms of the atomic resolution, we can just pick out one sort of little loop in terms of this. You can see that we can actually interpret it very nicely in terms of atoms. And so kind of technically it's working very well. The scientific question that was around this project is actually quite interesting. These holes over here have to accept the substrates. But the strange thing is that there's a converting enzyme that sits inside this kind of shell that looks almost like a virus. So the protein that sits in the inside actually does the conversion, does the catalytic conversion. But the strange thing is that in some cases the substrates are too large to actually fit through this hole. And so somehow the substrates are getting converted by the enzyme, but there's no apparent way that they can access them. And so we can visualize, exactly how big the hole is. We can visualize how big the substrate is, and we can also try and figure out how these kinds of things happen. And so what we always try and do, especially with the research that's actually happening within the EM unit, is that there's this very strong connection between the functional assays and the structures. The point of determining the structures is to try and figure out how these proteins function. And so it's very important to do experiments in both directions. So the structural work should inform the catalytic assays and the other functional work, and vice versa. Okay. And I just put these slides over here just to show that even though we are doing high resolution work, much of the work that we do is not necessarily high resolution, because you can get the scientific answers that you need in some cases without having to go to high resolution. It depends on what the question is. So with this little cage over here, the question here is the cage assembling properly? Are we expressing it nicely? Is the protein precipitated, or is the protein nice and functional and kind of structurally correct? And so at low resolution, you can see that. In terms of these things up here, this is spike protein, and you can see that the spike protein is nice and discrete, and it's being expressed properly. It's folded correctly, all of that kind of thing. So especially for screening, you would often not go to very high resolution. These are experiments that happen very, very quickly. So a reconstruction like this with a good protein sample, you could do easily within a day. The actual data collection can take something like 2 hours, and then you can process for an hour or so, because they're very small kind of data sets. So we're literally talking about just a few hours of work. And then once you put that work in, then you see more or less kind of the shape of it. To go to high resolution, it's months of work, or in some cases years of work. But certainly for low resolution, it's very, very simple and quick. Okay. So this is just more density and how it's been built. And this has become more and more common. So with a good protein sample, we can reliably get to this kind of resolution with an international data collection. And that's really one of the points that I want to make, is that we have got where we are because we access international facilities. And I think it's important to do so, because we're never going to be able to maintain the cutting edge in South Africa. Sorry, let me just go back one, like that. But we also can't get left behind. And so we need to have the technology to access international infrastructure. If we fall too far behind, we won't be able to put in successful applications for actually accessing this technology. And so one of the big risks that we have in South Africa is that with a single instrument going down or something like that, something breaking, we can literally have everything that we've been building up over the last however many years just immediately falling down to nothing, because if you don't have an instrument that you can do adequate screening on, then you can't access the international facilities, then you can't collect your data anymore. So this has become quite common for us, but of course it's always at risk. Okay, this is an experiment that's this is kind of an interesting experiment. So one of the things that we're doing, this is in terms of low resolution work, is looking at proteomic samples. And so the sample preparation over here is quite nice, because you can take, in this case over here, you can take bacteria, you can apply something to them so that they have some sort of physiological response. And once you've had that physiological response, you can analyze them in a variety of ways, including mass spec. So you can see differences in the proteins that are being expressed, the proteins that are present in the cell. But you can also do electron microscopy. So you can take the proteomic sample, instead of purifying each protein, you can just take a mix of proteins and simultaneously reconstruct a variety of different proteins. So this is a technique that I've become quite enthusiastic about, and I'd like to push it a bit further. I think it solves one of the problems in cryo-EM, which is good sample preparation. If you can use proteomic samples, it means that it opens up the door to a lot more samples, and it makes sample preparation very, very simple. So here are the resulting reconstructions. So this is with a single sample of a bacterial cell, and you can just basically see the protein complexes that exist inside the cytoplasm. So just to get back to what I was saying before about accessing these international facilities, I mean, that is an important thing. A lot of investment has taken place overseas in terms of investing in the best facilities and the best equipment and the best microscopes. But if we look at this map over here, we see the distribution of high-end microscopes around the world. And there are individual labs in Europe and America that have multiple very high-resolution microscopes. So the kind of microscopes that we can only access by going overseas. And you'll notice that in Africa, the entirety of the African continent, there is not a single one of these microscopes. So any person in Africa who wants to do any kind of high-resolution cryo-EM has to travel elsewhere in order to collect the data. Now that has worked for us so far, but of course if we want to expand this kind of enterprise and get more universities involved and more researchers involved and get more projects done, the reality is that we're going to at some point need to have a high-resolution cryo-EM somewhere in Africa. And I think the logical place for that is going to be in South Africa. And even in terms of South Africa, I think the logical place is going to be at UCT, because we have the staff and the facilities and the experience to do this. And what I've had, so I've had discussions with a funder who's agreed in principle to fund people to come along and use the instrumentation when it's in Cape Town. And I think that's really important, so that you can say that somebody who's working at WITS, who produces a sample, somebody's working in Pretoria, somebody's working in Durban, anywhere in South Africa, they can actually be funded to come down to Cape Town and have the microscope time be free, have their accommodation and their living expenses free to actually use this type of instrumentation. So I think that's a crucial step. The big problem that we're facing is that we're unable to actually fund the microscope. So that's really the greatest challenge. That's what we're trying to do at the moment. Okay. So That's just to make that point again. The second point here is that every project that we propose has to be approved internationally. Now, currently that's fine because we're coming up with projects that everybody agrees with. But as soon as priorities change, we could kind of see that with COVID. As soon as COVID happens, potentially COVID is a huge problem in Europe and America, and all access is then rerouted and it's devoted to only dealing with that one problem. We can have different problems, and we clearly do have different problems. There are different diseases that affect Africa. There are different problems that people in South Africa have, and the priorities are different. And so one of the issues with always getting prior approval kind of internationally is that kind of other people are assessing things based on the issues that they think are important. And our issues may well be different. Okay. So the facility that we would like to put together, this is a kind of a dream. We would like the possibility of people coming to this facility where they can use this high-end cryo-EM and they could get results, but that they don't need to become specialists. They don't need to become specialist cryo-EM people. So we're talking about automated data collection, and we're talking about assistance with processing. We would like it preferably to be free to access after a peer review system. Now that, of course, requires funders to get on board with that, which is potential. It can potentially happen, but we of course don't have the microscope yet, so we haven't been able to get anything on paper. We've only got verbal agreement to this. And our weaknesses are really in terms of protein production. So the facilities and the staff to produce good quality, very pure protein. The administration would need to, of course, be expanded in terms of getting as many people as possible to use this microscope. So billing, logistics, and reporting, which we're quite strong on at the moment, but of course this will have to be expanded. And the other problem is that these projects are incredibly computationally intensive. And so they can require weeks or months of powerful computing processing. But I believe that given time, the problem solves itself, because the resolution that we want to achieve stays constant over time, but yet computers improve all the time. And so if you have a more powerful computer, this will go more quickly and computers will become cheaper, and it will be fine. Because currently this is one of the bottlenecks. But I believe in something like 5 years, this problem will have solved itself. I mean, in order to achieve this, of course, we need large-scale national buy-in from people who are interested in doing this kind of work. So people who want to do these kinds of projects. And the sort of model that I like to look at this as is something like a synchrotron light. So it's a facility that people can come to, and there's a lot of action going around there, researchers assisting one another, or at least having discussions with one another, but not on the same scale and the same expense as a synchrotron. The importance of cryo-EM. It's predicted to overtake X-ray crystallography. I think the prediction at the moment is about 2024 in terms of the number of structures solved per year. The structures do take a bit longer to actually go through and solve, of course. But because you don't need crystals, it makes things a little bit easier. And so, and so there are advantages and disadvantages if you compare EM and X-ray crystallography. In some cases, X-ray crystallography is, of course, very important. It'll be probably the dominant technique for a while, but EM is certainly very, very close at the moment. And this is the last thing, it's just some acknowledgements to funders and people that have worked on these kinds of projects. And I'm going to stop sharing.
Thank you very much, Jeremy. I was supposed to introduce you, but then you just started talking and didn't get me. You just carried on talking. Anyway.
There we go. Sorry. Yeah, I can hear you. Sorry.
I see. So you turned off your loudspeaker. So
my speaker was off.
Okay. Thank you very much for the talk. It was very interesting. I'll just ask a quick question. Are you still servicing the need to do high pressure freezing and cryo-embedding for larger cellular structures? And isn't that a large part of the future?
Yes, of course, yeah, for sure. So we are, and in fact, internationally, it's one of the areas that people believe is the most important in terms of cryo-EM, because it allows you to see sort of structures in their context within the cell, and we of course have the equipment. One of the technicians is trained in this. We are doing it, but the problem is that in order to extract the data from these kinds of samples, we need to reliably collect tomography data, and that's really one of the strong reasons why we need to upgrade the microscope systems that we use, because the newer microscopes can more reliably collect the tomography data for high resolution data collection. That at least will mean that we can have preliminary data to then access international facilities. Because currently, realistically, we're not getting the resolution and the quality of data that we need to actually access those facilities. And hand in hand with that is sample preparation. So in terms of the sort of 5 to 10 year vision, the idea is a new cryo-EM, so a modern cryo-EM, and a modern FibSem. So a cryo-FibSem would then allow you to prepare these kinds of samples. You do high pressure freezing, put it into the FibSem, and you can fib out a nice small section in order to do tomography.
Yes. Okay, anyway. Okay, so anybody else want to ask a question of Jeremy?
I have my hand up.
Okay, go for it.
So hi, so I wanted to ask about the ACE inhibitor work.
Yes.
Now, so are you, in as much as you can divulge, are you carrying that work forward, and are you think you may be able to commercialize lisinopril that might be applicable on the continent for the population?
Yeah, so that work is going forward. There is a professor at UCT whose research interest is in terms of angiotensin converting enzyme, and what he's actually looking for is domain-specific inhibitors.
Yeah,
and so he's really kind of his sort of research aim is to try and figure out whether or not you can make a angiotensin converting enzyme inhibitor that's domain specific in order to reduce some of the side effects, and also in terms of kind of getting other effects. So that is really the goal of that work. In terms of moving forward with it, I guess the next thing would be to put in a nice inhibitor and to do another cryo-EM structure. The only issue is that the cryo-EM structure is technically very challenging to get. just because of all the flexibility.
Can I make a comment, Lawrence?
Yeah.
Jeremy? One of the very interesting things observed in that structure was the proportion of molecules that had actually formed dimers. Now, the dimers turn out to be inactive. So inactive at cleaving angiotensin. So the trick for a blood pressure controlled drug might be to work out how to make dimers form and inhibit them that way, rather than by using these rather nasty drugs that have horrible side effects. That was an interesting insight that came from that study, which was not really totally anticipated. Wouldn't you say?
What do you think? You can do something that modifies local salt concentrations or something like that?
No. Put another thing that sits in the dimerization interface that makes it bind tighter together. So all of these things are very subtle. They're not. Even the inhibitors don't bind all that strongly, and so it's a very subtle business, this blood pressure control.
What would you think is what role would this is a somatic ACE, right? What role would that play in terms of long COVID, which might be getting more into pulmonary physiology and not structural biology, but in as much as you could say anything, what do you think about that?
I have no idea. I've divorced the two different domains of study.
Wow.
And I mean, there's the interesting thing is that the other thing that has come out of all of these studies on ACE, and it must be said that structure, that full somatic ACE structure, was the first time that we've actually observed the glycosylations. We've actually observed the glycan side chains. But the thing that we'd worked out previously was that it was the actual vibration of the molecule that opened and closed the active site. And the drugs bound in, and they bound in differentially because of the different vibrational modes in each of the different subunits. And now we'd never actually been in a position to compute the vibrational modes correctly, but we observed them directly in the cryo-EM structure, which was terribly exciting. So a huge amount came out of that structure. It's a wonderful paper. Please read it.
Okay. All right. I have another line of inquiry if there's no other questions, then you got time.
Great, perfect.
Okay, so now getting back to the center and the kind of facility that you want.
Yeah.
You want to upgrade the current microscope that you have.
Exactly, yeah.
And this is, I guess you want to get to Glacios, or you want to go all the way to Cryos?
Glacius.
Okay. And they run like what, 10 million each? Or 2 million US dollars?
In terms of rands, it's it'll be about 80 million rands now with the exchange rate. What is that in US dollars? That is.
Let me calculate, because there's always something going on in the market.
It's 4 million.
4 million US dollars?
It's about 4 million US dollars, yeah.
But did you read PNAS? Did you read PNAS yesterday?
Nah, I'm behind on my PNAS reading.
Lawrence. Okay, so Russo at the MRC labs in Cambridge has published the design of their new poor man's cryo microscope, which is not yet been delivered to a fabricator, but it has very interesting features. It's 100 kV, they've made, unlike the Tundra, they've made a custom 100 kV gun, and they've made a custom 100 kV camera. And indeed, they've completely changed the cryo system. So it's an extraordinary machine, and it is going to put, well, they said at $500,000 would be the price of one of these instruments were to be fabricated by one of the major microscope manufacturers.
That sounds a little like you get what you pay for, but but I haven't read the paper. so I mean, that's quite a reduction in costs.
Yeah, yes, and indeed for single particle work, it seems that if you listen to what Jeremy said, most of the structures are not don't need to be solved at super high resolution to get the insights that are necessary. So this will be a machine that will give you single particle structures at $500,000.
The problem is that it prevents you from doing tomography.
The problem is that there are a lot of things that you can't do with it, but it's a specialist machine for doing single particle work. That's for sure. All it, it's not a fancy microscope that's going to sing and dance for you. It's simply a machine for doing single particle work.
For sure. Sounds like well, problem is getting even the $500,000 out of the government, right? So our biggest problem is how do we convince government that structural biology is worth investing into? Which so I mean, is there an argument, a successful argument to make that say that South Africa could make with itself and other governments on the continent or other governments in, like, say, in BRICS, and say, why don't we all buy a microscope and we can get some economies of scale?
Yeah, that's an interesting question. I mean, I heard that the Chinese have recently bought 75 Titan cryosys.
That's a lot of cryosys.
Yeah.
And think how the facility for manufacturing them would be completely log jammed. Nobody else in the world would be able to buy them for years.
Yeah. So the Chinese are ordering 75. So in terms of BRICS, they can just send one of those over.
we can all go to China.
Oh, yeah, right. You can all go to China.
Of course. The issue is that you know, some samples can't be transported,
right?
And some scientific work won't be funded there, and whatever. I mean, so in other words, it's important to have something geographically located in Africa, especially with things like agricultural viruses, which, I mean, you can easily kind of transport within the continent, image and get kind of great results, but you could never transport those things without sort of special approvals.
It's like smuggling in horse sickness virus into Frankfurt.
Yes, exactly.
Or even just beyond the biological arguments, but just customs and visas.
Yes, exactly, yeah, for sure.
Exactly. We have a colleague that is at the University of West Indies, that's in Barbados.
Yeah.
Long story short, she has been stuck here in the United States for a number of weeks, separated from her samples, and nobody knows nothing.
Yeah, it's crazy. Yeah, for sure.
Yeah, that might be something. You know, the African light source, we do talk about these sort of regional infrastructures or infrastructures. Yeah. We have looked at these sort of compact light sources, and I always try to argue, like, you know, cryo-EM is part of, well, I guess there's acceptance. I mean, we have a storage ring mafia, but there's there's some acceptance of cryo-EM as you know, part of local infrastructure.
Yeah, for sure. I mean, internationally, they're often located at synchrotrons because there are these similarities. It is this kind of facility that a lot of people can come to. They can bring their samples. They can do data collection.
Yeah, we got to have a lab space and the pipettes and all that stuff to even go to the whatever, beamline or microscope room. It's like the end of the beginning. The start of the nightmare.
Okay, I think if there are no other questions from anybody else, can we wrap up?
Okay.
Can I perhaps just ask a quick question? Sorry for joining late. I had connectivity issues on this side, but it's interesting. So what is the market like for getting a second-hand cryos, perhaps? I mean, these have been around for a while now. Kind of, aren't people switching to newer models and potentially looking at giving away their old ones?
Yeah. So the problem with the first generation cryos is not particularly reliable. So the reason that people, in some cases have upgraded and gotten rid of them is because of the extensive downtime. And that's even true of, to some extent, the Glacius 1, because now, so the instrument that's available is the Glacius 2. The Talos Arctica, which was the model just before that, there is a second-hand one actually available. So that has been offered to us by the agents, and the price for installation and everything else would be 30 million rand.
Okay.
So you're talking about less than half of the Glacius 2. The problem is that this level of instrument always sold for half as much as the Glacius in any case. Okay, so that's the first problem. And so the first generation Talos Arctica, which is what it was, cost half as much as the Glacius 1. Now they're trying to sell it to us for half the price of the Glacius 2. And this particular instrument apparently was down about 50% of the time.
Okay. Okay, perhaps not the best option.
Yeah.
So
But there must be others that will be coming onto the market. I mean,
For
even
sure,
if
for
this
sure.
is perhaps not the best, but perhaps.
So
maybe-
what I kind of showed on that other graph in terms of the instruments that we've kind of historically had is that very often the instruments are bought when they're something like 10 to 15 years old, and we then keep them going until they're something like 20 to 25 years old. Now, it has been a successful strategy because, of course, especially if you're just screening, that works. I mean, you get the data that you need. The problem is that the technology has advanced quite quickly. And so if you're always dealing with 20-year-old technology, I mean, 20 years is actually quite a long
I think
time.
the main thing is really to get an instrument here and then really demonstrate how productive it can be. And then I think government will kind of once the community really grows, then you can at the moment, the problem is that too few people in South Africa are using it. And then the government, everybody is just scared and says, no, it's and they just don't see the value in it.
Sure.
If you really.
So the reason that people are not using it is because there isn't one here.
Yeah, exactly.
I,
And especially if we can do things on African viruses, etc., I think that will, even if it is not the government, if it's industry, etc., I think they're potentially there's such a big market out there, kind of to get other players in the field, and not just universities, but industry and other groupings kind of involved in the whole process, and then it might be easier to actually collect the money from various other sources. I mean, the agricultural field, etc. I mean, that is what a lot of the funding is kind of coming from here at the university. Lots of the Millie board and the sugarcane board and the avocado board and all these things. They have they have certain amounts of money. And
of course.
Kind of get that.
It's a great tool for developing vaccines, or for developing, you know, some kind of
Or
treatment.
diagnosing plant diseases. South Africa agricultural kind of produce is a big market. So yeah.
I mean, what COVID showed is that whatever happens here is also going to happen there, you know, eventually. And so the argument that I've kind of been making is that, you know, it makes sense for the international community to invest in something like this, because you want instruments to be geographically spread around, you know, just from the point of view of planet Earth, you just want them to be around. And you
But
want people
the more people we can get involved, the smaller the amount that each one needs to pay, the kind of to get that.
Yes,
Because
yes,
again,
exactly. And if they're geographically far apart, then you have people working on individual problems. We work on the problems that are important to us: yellow fever, malaria, TB, these kinds of things. When those things become a problem overseas, then at least there's somebody working on those things.
Yeah, but then other people take over, and then they don't care whether it's done here or not, kind of thing.
No, look, I mean, that's fine eventually, you know, that's fine. But some kind of emerging virus, something like African horse sickness virus.
But if you get enough buy-in locally, I think that's always going to be the strongest card.
Yeah, exactly. But it's difficult enough to get people to buy into structural biology. Never mind a very expensive flavor of structural biology.
Yeah. But I think the trajectory is okay. It's going to, I'm sure it will work in the next 2 or 3 years or so.
We'll see. We're pushing hard.
You don't need experimental techniques anymore. You just got AlphaFold, and it's all in the database.
Yeah, of course.
Okay, let's wrap up. Thank you very much, Jeremy. That was very, very good. Thank you.
Cool. Cheers.